Myself, Dr. Ritchu Babbar, Associate Professor, Chitkara College of Pharmacy. Today I am going to discuss the topic anti-malarial agents, which is included in the syllabus of B.Pharm 6th semester, that is medicinal chemistry. So let us start with Good morning. Myself, Dr. Ritchu Babbar, Associate Professor, Chitkara College of Pharmacy. Today I am going to discuss the topic anti-malarial agents, which is included in the syllabus of B.Pharm 6th semester medicinal chemistry. So let's start with anti-malarial agents. The contents which I am going to cover in this, they are introduction, life cycle of Plasmodium, chemotherapy, that is the treatment, classification, quinine, about 8 aminophenolins, about 4 aminophenolins, bigonides, sulfonamides, artemisinin and its derivatives. Now let's start with anti-malarial agents. So let us start with the definition. What are anti-malarial agents? So the definition is clear to everyone that these are the agents which are used for the treatment and prevention of relapses of malaria. So before going to the topic anti-malarial agents and its classification, about the drugs, about the chemistry, let us first discuss in detail about what is malaria. So just see, malaria is formed by the combination of two words, that is mala and area. Mala means bad and area means air. So what is the basic meaning of malaria? It spreads in those areas which contain this bad air. So I hope everybody would understand bad air in those areas which are having poor sanitary and hygienic conditions, or whenever there is a stagnant water. Stagnant water, that is the basic reason, because the breeding of the mosquitoes that takes place at a very higher rate in stagnant water. So practically this malaria is spread by the bite of a mosquito. And specifically, the mosquito name is female Anopheles mosquito, that I have written here. Female Anopheles mosquito and that is your male Anopheles mosquito. Practically male that will feed on plants and female Anopheles mosquito that feed on blood. So this is the reason that why malaria would take place, because of the bite of this female Anopheles mosquito. But what else is the causative microorganism? It is the Plasmodium. I hope you would understand what is Plasmodium. Plasmodium is a protozoan. If I break the term protozoan into proto and zone, meaning proto means first and zone means animals. So that means this Plasmodium is a protozoa. Actually it is not a prokaryote, it is falling under the category of eukaryote, and it is a parasite. Parasite means which definitely requires a host for its living. So this parasite is having two hosts. What are the two hosts? One is the vertebrate. In vertebrates, animals, human beings, both are included, and definitely the animals are also more likely to be infected by this microorganism, that is Plasmodia. And second is the female Anopheles mosquito. Practically, female Anopheles mosquito is the carrier of the or the transmitter of the disease, that is malaria, and the life cycle of Plasmodium that requires two hosts. So in the next slide, I will be discussing detail about life cycle, because without life cycle, the chemotherapy or the treatment that would not be clear to you. Now moving on to this part, that Plasmodium species which affect the human beings, that because we are more concentrated about human beings, I am not discussing about animals. So they are the 4 species of Plasmodia which is more likely to infect humans. First is Plasmodium falciparum. So Plasmodium falciparum, it is the Plasmodia which is the most dangerous form. Why most dangerous form? Because this particular Plasmodia that develops resistance very rapidly. That means The person who is infected by this Plasmodium falciparum, that is unaffected, or that is not being practically treated with anti-malarial agents because they are not being effective. Now that causes malignant tertian malaria. Tertian means the cycle repeats after third day. I'll let you know what is the meaning of that tertian during the cycle when I'll explain that life cycle of Plasmodia. Then the second species is Plasmodium vivax. That causes benign tertian malaria. Again, tertian means the cycle repeats after third day. And Plasmodium malaria, that causes quartan malaria. Quartan means the cycle repeats after fourth day. And all the three. Next is the Plasmodium ovale, that causes rare malaria. Rare means very rarely we will finding the human beings which are being infected by Plasmodium ovale. Out of these four species, Plasmodium falciparum, that is the most dangerous, and the human beings are more likely to be infected by this kind of Plasmodia. Now the rest three forms, they are milder, but falciparum is dangerous. Now moving on to the important part, that how it requires two hosts, that is the human being and the mosquito. Now life cycle of Plasmodia, that includes mainly three stages. I hope the stages, they are clearly visible to you. First is, I will just read it out. Pre-erythrocytic phase, or it is primary exoerythrocytic phase. Second is secondary exoerythrocytic phase, and third is the erythrocytic phase. Before I will not discuss here about the stages, let us move on to the cycle. Now I hope this cycle is very much clear. Here the Plasmodia, because I told you earlier that malaria is a disease which is caused by the bite of female Anopheles mosquito, which I have shown here. This is the mosquito that practically contains Plasmodia in its salivary glands. So Plasmodia, they are in the form of sporozoites. Once it bites the human being, then it definitely transfers the sporozoites into the blood of the human being. And when this blood is infected by sporozoites, because these are mobile, so definitely they are going to move inside your body via blood. So what is the next stage they are going to move? They are moving within an hour. I repeat, they move within an hour, that is around 30 minutes to 60 minutes. They travel to the liver. So liver is the first organ which is being infected by Plasmodia, or it is sporozoites. Clear? Now from inside the liver, what else they are doing? Practically they are growing. Development and growth, that is being taken inside the human being, and first stage is the pre-erythrocytic phase, or it is primary exoerythrocytic phase. Pre-erythro, just break the term. Pre means first, erythro means before, erythro means the second stage would be when RBCs, that is the red blood cells, are to be involved. So practically in pre-erythrocytic phase, what is happening? These sporozoites, they get mature. Mature means they are taking food from the liver and they are getting mature into tissue size zones. And tissue size zones, they are definitely bigger in size than that of sporozoites, and ultimately it will reside in the liver for about 5 to 6 days. After 5 to 6 days, it is going to release that tissue size zone I am talking about, that releases thousands of merozoites. So I hope you would understand, if suppose 3 or 4 sporozoites they have been entered in your body, then they get multiplied into 1,000 times. Fine. Now these merozoites, that is the infectious form, it is having two ways. First way, see the arrow. First way, that few of the merozoites. So suppose if I am talking about 100%, so 100%, out of 100%, only 30% that remain inside the liver, and that will infect more liver cells, but they remain dormant. Dormant means they remain inactive. They are not going to infect you more by entering in the whole cycle. They just keep calm inside the liver, and this is the most dangerous form. Out of the 4 species of Plasmodia, only Plasmodium vivax and Plasmodium ovale, these two are more likely to become dormant, and these dormant species causes the relapses of malaria. So in the definition I have mentioned this thing, these are used for the treatment. Rather I would have used the word they are used for the treatment of malaria, but else I have used prevention of relapses of malaria. This is the basic reason, because these merozoites, they remain dormant in the infected human being, and they are likely, whenever they get the environment suitable for their growth, then they are likely to grow, and then relapses of malaria would occur again without the bite of the mosquito. Yeah? So for that also we need to take medicines so that that relapse of the malaria that won't occur again. Now where were we? About the dormant form. There is a specific name for the dormant form of your merozoites, that is known as hypnozoites. I repeat, hypnozoites. So these hypnozoites, they are the inactive forms and they are remaining inside the liver. Now what else about the active form? This is the active form in the pre-erythrocytic phase, that is the merozoites. I told you about if 30% they are remaining inactive, then 70% they are moving forward in the cycle. Now what is the second stage? This round the circle that represents the erythrocyte. Erythrocyte means it is the RBC. So definitely these merozoites, they are entering inside the RBCs and they are taking now food from RBCs and getting mature. Now in case of liver, we discussed about that sporozoites, they are getting mature into merozoites. Now it is the advanced stage that merozoites has entered the RBC, and this stage is known as erythrocytic stage. Clear? I repeat, the first stage was pre-erythrocytic phase, or it is primary exoerythrocytic phase, and second, inside the liver only, the dormant stage, that is known as exoerythrocytic phase, or it is the secondary exoerythrocytic phase. Now the third phase we have entered, it is erythrocytic phase. In erythrocytic phase, just see what is happening. This merozoid, they are getting matured, and the term which we use for the matured merozoites, it is trophozoid. So trophozoid, that may release further many merozoites that infect the human being. Practically, what is the common symptom for malaria? Everybody knows, that is the fever and chills. This is very important, that fever and chills, why do they occur and when is the symptom occur? Symptom occurs at the erythrocytic phase, when that erythrocytes contain so many of merozoites, and when these merozoites release, one thing more happen, that causes the rupturing of the RBC. So rupturing of the RBCs means that causes fever and chills, and at this stage we come to know. Before that, when the sporozoites, they have entered, they have been living in your body for the last 10 days, they are moving into the stage pre-erythrocytic phase and then secondary exoerythrocytic phase, that is asymptomatic, means the patient is unaware that this dangerous plasmodium that has entered in his body. But when do we come to know? When there is rupturing of the RBC that would take place. Now this rupturing of RBC releases so many of merozoites, and that causes fever and chills. One more thing I would like to explain here, because many students, they get confused about a term, that is a hemozoin. I just write on the blackboard, you just see hemozoin, because this is an important term related to the chemotherapy as well. What is hemozoin actually? Whenever this, let me write, see, when this merozoid that has entered the RBC, so definitely it would be taking food from the RBC. So RBC से food वो कैसे लेगा? From hemoglobin, because RBC contains that red pigment, it is hemoglobin. So it will break that hemoglobin into heme and globin portion. So heme portion and globin. Globin is what? That is the amino acid, it is a protein. So it will take amino acids from the breakdown of globin. But what else about that heme portion? That heme portion is actually toxic for the malarial parasite, that is the Plasmodium. So what will it do? Otherwise, if it is not doing anything for that toxic heme, the malarial parasite would die of itself. But what else it is doing? It is very smart. It is doing the polymerization of toxic heme to non-toxic hemozoin. So what it is doing? Heme, 1 molecule, 2 molecules, 3 molecules, 4 molecules, they are going to join to form a polymeric form, and that polymeric form of heme is known as hemozoin. And many students get confused about this word hemozoin, because which is quite similar to hypnozoite that we have used in the secondary exoerythrocytic phase. So after that, the cycle is not completed yet. Merozoites, they have been released and they are going to infect more RBCs, and more RBCs are infected, more trophozoites are forming and more rupturing. That means the condition gets worsened. The person is taking or having that chills and fevers after every third day. That means in the erythrocyte, this cycle would takes place around 1 to 3 days. Now moving on to another point, that if the merozoites or some of the merozoites, they are getting converted into their, or they are entering in the sexual cycle. This is the asexual reproduction, in which merozoites have been converted into mature form, trophozoite, and then it is more merozoites are forming by the rupturing of RBC. Simultaneously, the formation of the hemozoin. Now, these two are called as male gametophyte and female gametophyte. This is very important point, that these two, they will not get fertilized in the human blood. Reason being, there are not favorable conditions, might be some pH that would be required, some enzymes would be required for fertilization of these male and female gametophyte. Then what would happen? These two, male and female gametocyte, they remain as such in the body of the human being, but ultimately when the mosquito bite again the same infected human beings, then through the blood, these two, they are being transferred again to the mosquito. That's why I would use the word that there are two hosts for the living of Plasmodia. One is the mosquito and one is the human being. So till this, I hope it is clear about the cycle, that what else is forming in your body and what else, unnatural thing that is happening in your body that is causing problems to the human beings. Now, after that, what would happen? This male and female gametocytes, if the mosquito that sucks the blood from infected human being, then these two male and female gametocytes that enter in the mosquito, then where would they go? Definitely from the salivary glands, they will move to the gut. In the stomach, what else is happening? Actually, fertilization of male and female gamete that would take place, and that will get converted into a zygote, and after that zygote will release so many of sporozoites from where we have started. Now where these sporozoites will shift, not in the gut, definitely as soon as the sporozoites, they have been formed in the stomach of the mosquito, they will shift to the salivary gland or the in the mouth of the mosquito, and whenever it bites the healthy individual, it will start spreading or giving that sporozoites into the, or transmitting these sporozoites into the blood of the human being. So in this, how these are the stages that need to be discussed before discussing anti-malarial agents. I hope everybody would have understood, because there are so many sites where we can make our drug, where we can design our drug which will inhibit these particular sporozoites, merozoites, trophozoites and gametocytes. So let us again discuss about the chemotherapy. Before classification, we will discuss about chemotherapy, or it is the treatment. I hope everybody would understand why I am using the word chemotherapy. Chemotherapy is not actually the treatment with the help of chemicals. Actually, chemotherapy, we are using the word when we are more specific about the target. That means now we are having a target only with the Plasmodia, not with the human being. So that's why this treatment is known as chemotherapy. Although antibiotics, all are called chemotherapeutic agents. Cancer, because we are targeting dividing cells, so that is also a chemotherapy. Most of the students, they just think over that whenever we talking about chemotherapy, that are the anti-cancer agents. No, it is not like that. Chemotherapy is actually when we are targeting a specific target and not affecting the human being. Now moving on to the types of chemotherapy. I am moving along with the cycle also. See, here the first point is written that there is no drug available which act on sporozoites. Let us move on the cycle, that would be more easier to you for understanding. Now we can inhibit our first stage by making a drug which will inhibit sporozoite. If I use a word sporozoitoside, C-I-D-E, sporozoitoside, I hope everybody would understand. Sidle means to kill. So if we want to make a drug which is used to kill sporozoite, then that is known as sporozoitoside. So we can make a drug, but unfortunately in the market no drug is available which is inhibiting sporozoitoside. Then we have to search for other target. What would be the other target? If we go on to the second stage, that is pre-erythrocytic stage, or it is primary exoerythrocytic stage. Listen to me very carefully. The drugs which act on pre-erythrocytic stage, they are called as causal prophylactic drugs, because that is the root cause. Now, this is the first step where the sporozoites are going to divide and gets converted into merozoites. So if we remove the root cause, that's why they are called as causal prophylactic drugs. And definitely these are called as tissue scintocytes, C-I-D-E-S. First one was called as sporozoitoside and second is called as tissue scintocyte, because these are the tissue scintozones. Here also it is written very clearly that these are the sites for the action of tissue scintocytes. Now, second category of drugs are radical cure drugs. Radical cure drugs are practically used for the relapse or for the prevention of the relapse of malaria. In the first definition I have given that these are used for the prevention of relapse, because relapse is a major problem with malaria because of the presence of these dormant forms, inactive forms, that are hypnozoites that may get activated in the later in the lifespans, about many months, many years later. Fine. Now the drugs which will inhibit the exoerythrocytic phase, they are called as causal that is radical cure drugs. So you can learn this thing by R for R. R for relapse and R for radical cure drugs. Now we do have one more category, that we have inhibited two stages by our drugs. I will show you the classification as well. Now the third, we can inhibit at the erythrocytic stage. Erythrocytic stage means on the RBCs we can target our drugs which will inhibit the tissue scintocytes, or they are called as blood scintocytes. Why blood? Because we are targeting our drug to the RBCs which are infected by the merozoites. So many merozoites are formed in a single RBC, and you know the size of the RBC is quite small. Whenever there is rush inside the RBC, that will burst, and that causes the chilling and what else? That is the fever. So that's why we can make three targets. One is sporozoitoside, that was the target, but unfortunately there is no drug available in the market. But we do have many drugs which are inhibiting at the first stage, which are called as causal prophylactic drugs. We do have many drugs which do inhibit the hypnozoites, which are radical cure drugs. And third, we do have blood scintocytes, which is inhibiting the erythrocytic phase. Also, now the fourth or the last stage is what? Gametocytosides. We can also make a drug which will inhibit these male and female gametocytes. If we give the drug to the human being, then definitely these two gametocytes, they would get destroyed in the blood of human being, and whenever the mosquito bites, then these two are not being transferred to the mosquito, and ultimately no fertilization that would take place, and we would prevent the spreading of this disease to the mosquito. Obviously, mosquito is not important for us. What is important? That this, these gametocytes, they are not being spread by the mosquito to infect some healthier human beings. Clear? So I hope I have made myself clear with the cycle and their stages, and then obviously the chemotherapy. Now this is the classification. In this I'm going to study, this is the all pharmacology that we have discussed about the plasmodia cycle, about their treatment. Now we are going to start with the chemistry portion. So what else is the chemistry portion? Classification. First is the quinolines and their derivatives. Quinoline, I will let you know what is the basic nucleus quinoline look like. Under quinolines we do have three categories: 4-aminoquinoline, 8-aminoquinoline and quinoline methanol. Under 4-aminoquinoline, definitely the name indicates that quinoline is a nucleus on which the fourth position amino group is attached. I will let you know. The drugs are chloroquine and amodiaquine. Next is 8-aminoquinoline, that is primaquine and pemaquine. And third is quinoline methanol, which includes mefloquine. So these are the three categories which is coming under quinolines and their derivatives. Now second classification includes acridine derivatives. Acridine derivative includes the drug mepacrin, or the other name is quinaquine. Next is cinchona alkaloid, that means the natural drug which we obtain from cinchona bark. They are the alkaloids. It is quinine and cinchonidine or cinchonine. They are considered as the lead compound for this whole anti-malarial agents. I'll discuss about the lead compound as well later on. Now, moving on to bigonides. Bigonides includes chlorgonide, the other name is progonal, and fifth one is diaminopyridines, that includes pyrimethamine and trimethoprim, and then it is sulfonamides and sulfones, which include sulfadoxin, sulfamethopyrazine and dapsone. Then we have tetracyclines, which include doxycycline and tetracycline. This is a newer drug, that is artemisinin and its derivatives. Out of all the categories which I have enlisted, around 8 categories, only 2 drugs are there which we got from plant, that is the quinine, and the newer drug, it is artemisinin. Now I will let you know the reason that why do we shift on to the synthetic organic chemistry or medicinal chemistry, and why not we rely on the plant extract active principles. Reason being, there are 2 most important reasons that why every time we cannot rely on plants to get the active principle and for the treatment of certain diseases, because of the time. Because extraction from the plants, that would take longer period of time, and whatever the active principle we are getting out of the extraction, that yield is very low. So this yield is quite important. We want our drugs to be made at what? Higher rate or in the bulk. So for that we need to rely on the synthetic organic chemistry. And the third reason is the structures which we get from the plant, from the let it be alkaloids, let it be tritapenoids, they are quite complex. So we want to make, which are very difficult to synthesize. If we want to study synthesizing quinine in the lab, that is quite difficult. But if we want to synthesize a drug in the lab, that would have quite simpler structure which can be easily synthesized. Fine. Now moving on to first category, it is quinine and other amino alcohols. First we just look on this structure quinine, and here it is written cinchonine. Now listen to me very carefully. When this is the lead compound, I hope everybody would understand about the lead compound. Lead compound is the compound which is discovered first with pharmacological activities, and many students, they give the definition, what? The lead compound which is discovered first, but this is a big wrong. Until you use the word pharmacological activity, the compound is not considered as lead compound, but with some drawbacks, and these drawbacks need to be modified. And whenever we take the idea from the structure of the lead compound, then we could make many drugs by making general structure and by doing certain modifications which is causing drawbacks to the lead compound. We can omit that functional groups and we could add more functional groups so that we could get a potent drug rather than that particular lead compound. Clear? So this is the lead compound quinine, which was obtained from cinchona bark. Actually we got 2 pairs of isomers. 2 pairs of isomers means we got 4 compounds. 2 pairs means 4. One is quinine and another is quinidine, and the third is cinchonine and cinchonidine. Quinine and quinidine, there is a very small difference. Quinine is considered as a levoisomer and quinidine is considered as a dextroisomer. Similarly, cinchonine and cinchonidine, small difference. Just see what else is the difference between cinchonine and quinine. In quinine, there is a methoxy substituent which is attached at the 6th position. If I start numbering from here. Now, before that, I should tell you about quinoline. I have given quinine and other amino alcohols which were falling under the category of quinoline. So, this nucleus is known as quinoline nucleus, which is an aromatic bicyclic with a heterocyclic atom, nitrogen. So, we need to start numbering from here 1, 2, 3, 4, 5 and 6. At 6th position, we are having methoxy, but in case of cinchonine, there is no methoxy substituent attached. This is the only difference between quinine and cinchonine. But actually, what else is important for us? Out of the 4 isomers, quinine had shown maximum anti-malarial activity. That's why we have chosen this quinine. Although quinidine, if you must have seen that quinidine in the antiarrhythmic classification, so that will be discussed there. But now we will be discussing about quinine. So this quinine, moving on to the next slide, because this I will explaining the structure activity relationship, that what leads to the modification, that we need to do the modification in this lead compound to get more potent compounds than quinine. Just see, this is quinine and this is its dextroisomer quinidine. You can very well see the difference here. Here the OH is with the beta configuration and here the OH is with the alpha configuration. Means this OH is pointing towards us and this OH is pointing away from the observer. Now it is having, now I'm going to discuss with the chemistry portion of quinine. Why we need to do modification in quinine. One drawback related to quinine is it is quite bitter and everybody knows that it is a bitter compound. Also, that means the toxicity. So what else we need to take? We need to make certain drugs with lesser toxicity and less bitterness. What else is the problem with quinine? First discuss with the chemistry portion. It contains quinoline moiety, the encircled portion, and this bicyclic nucleus, that is known as what? Cyclic amine, or it is known as quinidine moiety. Quinidine moiety is this and this is quinoline moiety. On quinoline moiety, 6th position methoxy group is attached, and in quinidine, this vinylic portion that is attached at the 3rd position. Now what else? After that we need to see these two molecules, they are joined by a linker. So what is that linker? This linker is secondary alcoholic functional group. So what else is important? It contains quinoline nucleus, quinidine moiety. At quinoline we have 6th position methoxy and in quinidine we have a vinyl functional group, and both these are joined by secondary alcoholic functional group. Now we have to see, because it is having quite complex structure which is very difficult to synthesize. Now we will be seeing that what functional groups are essential for the activity and what are not. If the functional groups which are not essential for the activity, definitely we need to do modification. Either we need to delete these functional groups and add simpler functional groups. After doing extensive research on quinine, what the scientists would find, they found that quinoline moiety is essential for the activity. So what does it mean? We need to put quinoline moiety in our structure, but what else? This methoxy group, that is not essential. That means if we remove this methoxy group, then also the anti-malarial activity would be there. And second is vinyl functional group, that is also not essential for the activity. We can also omit that functional group. What is most important about this quinidine, which is a quite complex moiety? What they found, that this moiety is also not essential for the activity. But what else is important? Important is alkyl. This is the alkyl CH2, tertiary amine. This nitrogen is in tertiary position. What they found? This whole moiety is not essential, but alkyl tertiary amine, that is essential for the activity, and necessarily it should attach with the secondary alcoholic functional group. This was essential because they have removed this hydroxyl functional groups by doing esterification, like if I put OCOCH3, OCOC2H5, like if I do the esterification, then also the activity would be reducing. Or if I do the oxidation, like what else the product I am getting when OH is oxidized, that is COOH. So definitely if I am going to change or replace this hydroxyl functional group, then the activity would be reduced. So we need not to do such modification that may lead to less potent compounds than the already existing ones. Already existing one is what? Quinine. Now we want to make our drug to be quite simple than quinine and definitely more potent than quinine. Now what else is the general structure that we got after modification of this quinine? I am writing here, it is aromatic nucleus. Then we have secondary alcoholic functional group, which I just told you that this is important. What else is alkyl tertiary amine? So I hope everybody is clear about the point. This is the general structure. We want to make drugs which look like quinine but better than quinine, which can be synthesized easily in the lab. They should contain aromatic nucleus. From where I got the idea? Aromatic means I should be having quinoline specifically. I can write here quinoline also, because definitely you will see so many drugs that contain this quinoline moiety. Then secondary alcoholic functional group, I just told you that this is important, so I have to keep this functional group attached on the quinoline moiety. Alkyl tertiary amine. Alkyl, here I have written CH(N). That means the chain can be increased or decreased depending upon the activity. Fine. Now this is the tertiary amine. So what drug we got after modification or after this general structure? Just see. Just see this mefloquine. This is the synthetic drug which we obtain after doing or after getting the general structure which I have just written here. Just see whether all the compounds are present or not. This is the aromatic nucleus. So aromatic nucleus, that I told you quinoline is required. So we have put quinoline as such with the secondary alcoholic functional group which is attached at this position 1, 2, 3 and 4th position. And then I told you it is alkyl tertiary amine. Although this mefloquine contains, it doesn't contain tertiary, it contains secondary amine. But rest of the structure, that is according to the SAR. So this is the synthetic drug which we got after moving on to the general structure of quinine and cinchonine. Clear? So I hope this point is clear to you. Let us move on to the mechanism of action. So these are all the points that I have discussed just now and they are written in this. Mechanism of action of quinine, very important, need to be understood. I have given you 3 cycles. One is pre-erythrocytic phase, another is secondary exoerythrocytic phase and third is erythrocytic phase, or it is the clinical or suppressive cure drug. So quinine, it is a blood schizontoside. So I hope you would understand. Blood schizontoside means I just show you the cycle. That means it is acting on the erythrocytic phase, specifically very important, because if we inhibit at this stage, definitely the patient will get rid of the symptoms. Symptoms kaun se? That is fever and chills. If we gonna prescribe the patient the hypnozoitoside or the tissue schizones, definitely the division of the merozoites or the number of the merozoites, sporozoitosides, that would be decreased. But fever and chills get only be cured only when we have to give the patient blood schizontosides, and lot many drugs are included, not only quinine. Now that's why always anti-malarial drugs are to be given in combination, because when do we get the symptoms? We get symptoms at this stage. So in our body, lot many hypnozoites are there, lot many merozoites are there, and lot many again bursting of RBCs would be taking place. So we have to take combination of drugs, like for radical cure drugs we need to take one drug from tissue schizontoside and one drug from blood schizontoside. So when I am talking about quinine is a blood schizontoside acting on the erythrocytic phase. Now what is the problem? The side effects were synchronism. Synchronism means ringing of ears. not if you take this medicine for 3 or 4 days you will be observing these kind of symptoms, but actually when you are taking for longer period of time, then these kind of symptoms that would take ringing of ears, difficulty in hearing, nausea, vomiting, rashes. So for that we need to do modification and we have to switch over to other drugs which would be more potent than quinine, more easier to synthesize in the lab as compared to quinine. So I hope I have made myself clear with this first classification. These are the marketed preparations that we will be seeing in the market with quinine sulfate, which is a salt. Moving on to the second category, it is 8-aminoquinolines. 8-aminoquinolines, as the name indicates, the basic moiety it would contain is quinoline, which we got the basic idea from quinine. Because in quinine I told you that quinoline moiety is essential, so we will be keeping this moiety forward and getting more potent drugs. So definitely with the name 8-amino, at 8th position we would be having amino substituent. Now what is the lead compound of this category? Lead compound was methylene blue. I hope everybody is clear about the point. Methylene blue, which is a dye, and many years ago this particular methylene blue that is being used as a indicator, fine, but surprisingly it has shown anti-malarial effect. Definitely not more potent than quinine, but now our research is what we want, a drug better than quinine, so we need to do modification in the methylene blue, which we got just surprisingly. Just see methylene blue, we have to start numbering from here: 1, 2, 3, 4, 5, 6, 7 and 8. This is the 8th position where amino substituent is present, and that served as the lead compound methylene blue. It was less effective and toxic, so we need to do modification. But we got an idea that we can make the compounds which contain 8 amino substituent at quinoline moiety. Now what we found, just see here, modifications were done. But what was the functional group which was found to be active? It is dialkyl amino alkyl moiety. I repeat, dialkyl amino alkyl moiety. When we substitute on the 8th position of nitrogen, 8th position of quinoline with the amino substituent, like amino, if it is attached, NCH3, CH3 is attached, fine, in methylene blue. So one of the methyl substituent is being replaced by dialkyl amino alkyl moiety. What it was found? Surprisingly, greater or more potent drug that was found when we did modification in methylene blue. So after doing certain more steps, we came to know about a more potent drug. It is pemaquin. I repeat, what is the lead compound for 8-aminoquinoline? It is methylene blue. If we talk about quinine, so that is the lead compound for whole antimalarial. So you can write, if somebody asks you a question like write the detail about anti-malarial agents, then you can start with quinine as well, because quinine is the first compound which was isolated from cinchona bark. Fine. Now this is methylene blue. After that we got pemaquin, because we got the idea from your methylene blue lead compound that we can substitute or we can make such compounds which contain 8-aminoquinoline. I am starting numbering from here: 1, 2, 3, 4, 5, 6, 7 and 8. तो 8th पोजीशन पे आपको क्या मिला? दैट इज अमाइनो सब्स्टिट्यूएंट. और अभी मैंने क्या बोला था, जब हम अमाइनो सब्स्टिट्यूएंट में क्या करते हैं मॉडिफिकेशन, डाई amino alkyl moiety, when we substitute on the 8th position nitrogen atom, then we could get more potent compound. So this is the second thing that we got, and the basic moiety which is 6-methoxyquinoline, we got the idea from quinine. तो हमने दो combination से एक drug बनाई, कौन सी? Pemaquin. One, we got idea, the basic ring about quinoline from the quinine, and one, the side chain, we got it from doing modification in methylene blue. But sooner this drug is now not in market. क्यों मार्केट में नहीं है? क्योंकि इसकी टॉक्सिसिटी बहुत है, इसलिए इसको विड्रॉ कर लिया. तो इसके बाद मॉडिफिकेशन की गई. जस्ट सी द डिफरेंस बिटवीन द स्ट्रक्चर ऑफ pemaquin and primaquin. Prima, as the name indicates, it pyrimethamine. Then we got a new drug, it is primaquine. So I hope this difference is very much clear to you, that what is the difference between pyrimethamine and primaquine. Pyrimethamine contains diethyl amino moiety, and in primaquine all the structure that is same, only the primary amine is different, which is the most potent drug of this category. I am saying, I am not saying that it is the most potent drug of all antimalarials. Clear? This is the structure activity relationship which I have just explained. This is the mechanism of action, very, very important. Many times the question has been asked about primaquine mechanism of action as well as its discovery. Discovery means you need to start, you can start it from quinine as well, giving you the general structure of quinine, giving the drawbacks of the quinine, that why we need the modification. Then you can switch over to the methylene blue, which is the lead compound, and then move on to PEMA and then prima. Now I am telling about the mechanism of action. It is again what? It is not a blood cytotoxide. Although quinine was a blood cytotoxide, and what about the primaquine? It is acting on both the stages. कौन से वाले stages? Primary stage, that is pre-erythrocytic stage, and secondary exoerythrocytic stage. मतलब this is very much important radical cure drug which will inhibit the hypnozoid which is responsible for the relapse of malaria. तो अगर हम quinine ले रहे हैं तो हमें उसके साथ primaquine को भी लेना पड़ेगा, because this is having both the action. It is killing the first as well as the second stage. Second thing, how it is going to kill the plasmodial DNA? Plasmodia by inhibiting the DNA synthesis, and everybody knows that DNA is the main genetic material which is responsible for transcription and translation. If we are going to inhibit the DNA, definitely we are going to inhibit the whole microorganism. Fine. Also, very important step that I just explained, the difference between hypnozoid and hemozoid. This is inhibiting the polymerization of toxic heme to non-toxic hemozoid. प्लाज्मोडिया क्या कर रहा है? आपका हीमोग्लोबिन यूज कर रहा है अपनी ग्रोथ के लिए. कैसे? By breaking down the hemoglobin into heme and globin portion. Globin is being utilized by the plasmodia for its growth, amino acid, whereas the what else? Heme portion, that is toxic. What else it is doing? It is doing the polymerization by joining 1, 2, 3, 4 heme molecules to form a non-toxic hemozoid. So what this primaquine is doing? This primaquine is inhibiting that polymerization of heme, and that is toxic for again the plasmodia. क्योंकि जब हमने उसकी polymerization रोक दी तो इसका मतलब हमने non-toxic form में उसको convert नहीं होने दिया, and else the heme which is toxic, that will kill the plasmodia as well. Clear? So that's why its mechanism of action is very, very important. 2-3 steps we do have. Now it is mainly used as radical cure. Just I explained, because it is acting on secondary exoerythrocytic phase, so it is used as a radical cure drug, and it should be given with other blood cytotoxides like quinine. अभी तक आपने सिर्फ quinine पढ़ा, तो I am using the word quinine. We will be studying more blood cytotoxides as well. These are the marketed preparations of primaquine in the tablet form. Then moving out with the third classification, 9 aminoacridines and 4 aminoquinoline, which is very, very important. You must have heard about chloroquine, which was very popular drug few days back about the coronavirus, because some clinical trials would have done on this chloroquine, which was inhibiting the virus as well. Now this is the most, and listen to me very carefully, I am talking about chloroquine, which is the most potent drug of this category. Category means whole anti-malarial agent. What is that? Chloroquine, just see how it is being developed. It is being developed from 9 aminoacridines. तो पहले कौन develop हुआ था? 4 aminoquinoline से पहले 9 aminoacridines. Just see the structure. This is a tricyclic moiety, and the whole moiety is known as acridine nucleus. And its 9th position, it contains amino substituent. तो जो 9th position पे amino है, then it is known as 9 aminoacridines. Fine. उसके बाद, if you just see the structure, that it is having structural similarity with pyrimethamine and primaquine. Just see dialkyl aminoalkyl moiety. So half of the portion when we synthesized mepaquine or quinaquine, we got the idea from primaquine, and this acridine nucleus. Actually, there was some drawback of mepacryl, that it was staining skin yellow, just like tetracycline. When we take, no, then that causes the skin yellow, teeth yellow. So that is quite, this is visible with the naked eye to the other person. No? So that's why we need to do certain modifications so that we could get a better drug than mepacryl. So what, just see, if I hide this ring, because it is a tricyclic moiety, if I hide this ring, just see, can you imagine that it is a quinoline? It is just looking like a quinoline. And when I give the numbering 1, 2, 3 and 4, at fourth position what is having? Amino. If I hide from this side, then also 1, 2, 3 and 4. This is again a quinoline, this is again a quinoline. So can I say that acridine or mepacryl, that contains, or that is the combination of 2,4 amino quinolines. In this way, we could get an idea that when acridine is formed by the combination of 2,4 amino quinoline, now can we take that 4 amino quinoline as a basic moiety and will synthesize a drug that will show certain effect as anti-malarial. So this is the basic reason that how we develop 4 amino quinoline. That's why I am saying it is very important. If somebody asks you a question, write a note, draw the structure activity relationship of 4 amino quinoline. So always it is advisable that you start with lead compound. So the lead compound here it is mepacryl, which is an acridine nucleus formed by the combination of 2 quinolines. कौन से वाले 4 amino quinoline. Then we got to know about a potent drug which is a chloroquine. Now just concentrate on the structure of chloroquine. The structure of chloroquine includes this quinoline moiety. If I start numbering from here, 1, 2, 3, 4. At fourth position, I am having this amino substituent. So this is 4 amino quinoline with chloro substituent at 5, 6 and 7th position. With the side chain, we got an idea from primer. Fine. Now, I am just giving you the points related to the SAR. That what else is important for chloroquine? Quinoline moiety is essential. Dialkyl aminoalkyl side chain is essential. We should not remove this side chain, or we should not remove or replace this aromatic nucleus which is quinoline, which we got the idea from, you can say pemaquine, you can say quinine. Next is at fourth position, this amino substituent, this is essential. 7th position, chloro is essential. That's why its name is chloroquine. Chloro for chlorogroup, quin for quinoline. Fine. Now what else about the other positions which are unsubstituted? Definitely we should give the points also whether we can do substitution on these unsubstituted positions or not. So what they found, that 3rd and the 8th position, that need not to be substituted. इनको substitute नहीं करना है, rest को आप कर सकते हो. But अभी there is no drug available of 4 amino quinoline category which would be better than chloroquine. Clear? Moving on to the next drug, it is amodiaquine. Just see a very small difference between the structure of amodiaquine. Basic structure is it is 4 amino quinoline. Moving on to the structure activity relationship which I just explained. Important is what? Important is this mechanism of action of chloroquine, because somehow whenever you attempt the questions, different questions come depending upon the mechanism of action as well, depending upon the SAR. So what else you have to include in the SAR and what else you have to write in the mechanism of action, that is the important thing. So this is the thing that I just explained for pemaquine and primaquine also, that both are inhibiting the polymerization of toxic heme to non-toxic hemozoin. So similar is the case this chloroquine is doing. What else it is doing? Your plasmodia that will break this hemoglobin into amino acid and the heme portion. This red portion is the toxic form for the plasmodia, and it will convert it into hemozoin, which is the polymerized form, which is non-toxic for plasmodia. It can survive in the presence of hemozoin. But whenever we are prescribing the drugs, which drug? It is chloroquine, which is a blood cytosine as well, which is also inhibiting the polymerization of that is hemozoin. So this will inhibit the plasmodia in this way, that it will inhibit the polymerization of heme. These are the marketed preparations of quinaquine as well as chloroquine, that is 4 amino quinoline, as well as 9 amino acridines. Next category it is the bigonides and aminopyrimidines. Bigonides. I hope everybody would understand bigonides. See, whenever we did that chemotherapy, I need to show you certain things in that which I think it was skipped. See, the treatment. In this, I told you that there is no drug available which will inhibit sporozoid, and causal prophylactic drugs again primaquine, pyrimethamine. So this pyrimethamine is actually the pyrimidine derivative. Radical cure drugs again primaquine we have discussed in detail. This is clinical or suppressive cure drugs, that is blood size orthosides. It is having two categories. One is faster acting high efficacy. Definitely the doctor would prescribe you first these faster acting high efficacy drug, and if you somehow unfortunately get resistant to these drugs, because I told you in the start of the chapter, or that in the start of the lecture, what else, that Plasmodium falciparum that gets resistant very rapidly. So for that you need to switch over to the other alternatives as well. So faster acting, it is chloroquine. So chloroquine is acting under blood sizontoside. Although this is also a very important question related to GBAT also, that what are blood sizontosides, what are tissue sizontosides, what are merozoites and all. So chloroquine, quinine, mepaprin and mefloquine, these are faster acting drugs. Slower acting drugs which we are going to just start, that is bigonides. It is proguanil, pyrimethamine and sulfonamides, although they are blood sizontosides. Now moving on to guanide. Just see, bigonides. I hope the functional group of guanide, it is clear to you. What is that? When this carbon is attached with 3 nitrogen atoms, it is known as guanide molecule. And when 2 guanide molecules are present in a single molecule, that is called as bigunide. Just see, this carbon 1, 2, 3, 1 guanide. This carbon 1, 2, 3. This is common for both. So it contains 2 bigunide moiety. That's why it is known as, or that's why it is coming under bigonides. And that had also shown anti-malarial activity, but not better than chloroquine. Although अब हमारे पास वो ड्रग्स क्यों हैं जो कम पोटेंट है क्लोरोक्विन से, जो कम पोटेंट है क्विनीन से? रीजन सॉलिड इज व्हाट? Because of the resistance, emergence of resistance, we require multiple drugs so that we could save the life of a patient. So this is proguanil attached with the chlorobenzene. Moving on to next, that is the aminopyrimidines. Aminopyrimidines, I hope you understand pyrimidine. This is the nuclear 6-membered heterocyclic with 2 nitrogen atoms alternate. And here we would have amino substrate. Always this numbering starts from nitrogen. So 1, 2, 3 and 4. So this is 2,4-diamino pyrimidines. So these are the derivatives that contain the basic moiety, and this moiety was proven to be very successful anti-malarial drugs. Although they are having the mechanism of action common. क्या mechanism था इनका? Just like sulfonamides. If you remember, we do have a whole mechanism for folic acid synthesis. I will be discussing here. Pyrimethamine, trimethoprim. Just see trimethoprim. It also contains 2,4-diaminopyrimidine. Why this is known as trimethoprim? Just break the term trimethoxyprim for pyrimidine. So this is trimethoxy and this is pyrimidine. This is how its name is, trimethoprim, which is a dihydrofolate reductase inhibitor. I hope all the 3 drugs, proguanil as well as pyrimethamine. देखो pyrimethamine का name pyrimethamine क्यों है? Prim stands for pyrimidine, amine stands for amine, and etha stands for ethyl. Clear? So this is pyrimethamine and this is trimethoprim. All the 3 drugs which are low efficacious drugs, they are coming under blood sizontosides. That means they are acting on erythrocytic phase. It will inhibit the symptoms, that is fever and chills to the infected patient. See, the mechanism of action, this is structure-activity relationship which I just told you, that this guanidonucleus, that is essential for the activity. We need not to replace this particular nucleus if we want to make our drug under the category bigonides. And again, if you want to make the drug under aminopyrimidines, it should contain pyrimidine as the basic moiety with 2,4-diamino substituent. Fine? Mechanism of action, I just told you that they are dihydrofolate reductase inhibitors. Now the question arises that what is the importance of this enzyme? Definitely it is doing something with the plasmodia, and plasmodia requires this particular enzyme for its living. Then only this may be a target to kill the plasmodia. I just show you this. These are the marketed preparations. Aminopyrimidines, sulfamethoxazole and trimethoprim, which is the most common combination co-trimoxazole. This you must have studied in sulfonamides chapter also, and this is trimethoprim tablets specifically applied for the anti-malarial. Now coming to the less potent drug, it is sulfonamide, which is a blood isotoside with less efficacy. Just see the thing. Everybody knows that folic acid is essential. Why folic acid is essential? Folic acid is essential for the DNA synthesis, not only for the human being but also for the microorganisms. If I talk about bacteria, which are prokaryotes, definitely they are not going to synthesize their folic acid of its own. That means they are not going to take the folic acid from the diet. Rather, they are going to synthesize their folic acid of their own. वो कैसे बनाएंगे folic acid? With the help of these two enzymes, dihydrofolate synthase and dihydrofolate reductase. ठीक है? अब हम कैसे इन ड्रग्स से unaffected होंगे? If suppose you are taking sulfonamides, all sulfonamides are written here. They are inhibitor of dihydrofolate synthase. चलो पहले हम ये डिस्कस करते हैं कि folic acid बन कैसे रहा है. First two reactants that we require, it is pyridine residue and para-aminobenzoic acid, commonly known as PABA. When these two unite in the presence of enzyme, what? Dihydrofolate synthase, that will synthesize dihydroteroic acid. Dihydroteroic acid, by the presence of same enzyme, that is dihydrofolate synthase, it will synthesize dihydrofolic acid. Fine. Dihydrofolic acid is getting converted into tetrahydrofolic acid by the presence of enzyme dihydrofolate reductase, which is very important. अब ये tetrahydrofolic acid क्यों चाहिए? देखो रिडक्शन हुई है, रिडक्शन का मतलब क्या हुआ? एडिशन ऑफ हाइड्रोजन. पहले क्या था? डाइहाइड्रो, नाउ इट बिकम्स टेट्राहाइड्रो. So this is practically involving in the synthesis of DNA nitrogenous bases. Nitrogenous bases means purines, pyrimidines की synthesis में dihydrofolic acid is acting as a coenzyme. और अगर ये नहीं है, suppose there is a deficiency of tetrahydrofolic acid in your body as well, in the bacteria, in plasmodia, then what would happen? That will not lead to DNA synthesis. So that is quite fatal for microorganism. Same for the bacteria as well as same for plasmodia. अब हमने कौन सी drugs ली हैं? We have taken this sulfonamide as a target. We have taken trimethoprim or pyrimethamine as a target. What sulfonamide is doing? Sulfonamide is inhibiting the whole cycle, doesn't allow the formation of tetrahydrofolic acid by inhibiting the enzyme dihydrofolate synthase. And one thing I must tell you, dihydrofolate synthase is not present in human beings. So we are unaffected by sulfonamide. Suppose if you are taking sulfonamide, then that will not harm you. Definitely it will target for the bacteria or the plasmodia because this particular enzyme is not present in our body. Second thing, if we are taking trimethoprim and pyrimethamine, that is going to inhibit dihydrofolate reductase. Dihydrofolate reductase, which is doing reduction. Now the question arises ki whether dihydrofolate reductase is present in human being or not. Yes, it is present, but important thing is trimethoprim and that is your proguanil or pyrimethamine. These are the drugs which are having higher affinity for dihydrofolate reductase of plasmodia or the bacteria rather than human. So that's why human beings are unaffected and they are termed as chemotherapeutic agents. So what else we are doing? We are not allowing, these drugs are not allowing tetrahydrofolic acid to be formed inside the plasmodia. Then what would happen? There is a deficiency of this tetrahydrofolic acid which is acting as a coenzyme for the synthesis of DNA and ultimately the plasmodia would die. So this is the mechanism. So I hope this concept is clear to you that where sulfonamides and where dihydrofolate reductase inhibitors, they are acting in the cycle. Now comes the last category which is very important, that is artemisinin and its derivatives. So I told you in the start of the cycle that we got two drugs naturally. One was the lead compound of this category, which was quinine, and quinine we got from cinchona alkaloid. I mean cinchona bark, which is quinine is an alkaloid. Now this is artemisinin. Again artemisinin, that is, what is that artemisinin? We got artemisinin from Artemisia annua. That is the plant Artemisia annua, and bistructurally it is a sesquiterpene lactone endoperoxide. It is a quite long word. I should clear the point through the structure that what is sesquiterpene lactone endoperoxide. Just see this whole, the 3 tricyclic system, it is sesquiterpene. Lactone means when it contains OCO moiety in the cyclic ring. That is the ester moiety in the cyclic, that is called as lactone. Fine. This is sesquiterpene lactone and this is endoperoxide, just like H2O2. Fine. That is O linkage is endoperoxide. A quite complex structure. Again, I should use the word. Why? Because we are getting it from plant, and maximum plants, whatever the drug we got. Like if I give you the example for physostigmine, that is acetylcholinesterase inhibitor, which we got from plant, again a complex structure. We need to do modification to form simpler structures. Similarly, anticholinergics, about that atropine, again it was having a complex structure. Amitin for antihelminthics, that was also having a complex structure. And again it is quinine that you must have seen, and this is the artemisinin. So what was the good thing and what is the bad thing about artemisinin? Why we have made around 4 to 5 derivatives of artemisinin. Why? That means there would be some speciality in this artemisinin. That's why we have done modification. First, I'll let you know about its good thing. Then we'll discuss about its bad thing. Good thing is what? Artemisinin was, when we isolated it from Artemisia annua, what it showed? It has shown good activity against falciparum resistant, against chloroquine resistant falciparum plasmodium. मतलब क्या हुआ? जो पेशेंट्स क्लोरोक्विन से ठीक नहीं हो रहे थे, क्यों नहीं हो रहे थे? Because if the falciparum plasmodia that has become resistant means that is not showing activity, that is not showing effectiveness against that chloroquine. आप chloroquine की high dosing ले रहे हो, लेकिन फिर भी chills और fevers ठीक नहीं हो रहे. What is the meaning? That you are getting resistant to chloroquine. So what is happening? This artemisinin, it is showing benefit, or it was showing benefit to those patients which were resistant through that chloroquine that against that plasmodium falciparum. So this was the good point, that we need to do modification here, we need to do research on this artemisinin, because we got an beautiful or a good alternative to that, the most potent drug of this category, which is chloroquine. Fine. But the drawback, here I have written, drawback is what? It is having poor water as well as lipid solubility. दोनों ही चीजें इसकी poor थी. So we need to do certain derivatization so that we could get a better drug which would be having better water solubility as well as better lipid solubility. I'll show you कि क्या दिखाया. But before that, I'm giving you a highlight about the mechanism of action. Just see, it is the basic mechanism of action that resides in its endoperoxide linkage. That endoperoxide linkage, when it goes inside the parasite's body, then what else it do? It is getting broken down. Now, when this particular endoperoxide linkage is broken down, then that is forming free radicals. And always that free radicals that causes lot of damage to the proteins as well, lot of damage to the, what else? The DNA. So what else that artemisinin was doing? Artemisinin, when it is entering in the plasmodia, that will be broken down into free radicals because of the breakage of this endoperoxide linkage, and that causes the damage to proteins, damage to the DNA, which cause the killing of the plasmodia. अब हमें क्या करना है? We need to do the derivatization, कि जिसकी वजह से हमें प्रॉब्लम है. अब ये तो हमें पता चल गया that artemisinin is very good drug which is even showing good effect against chloroquine resistant patients. ठीक है? तो we have done modification. जैसे हमने कहाँ modification की? We do have 4 derivatives: artemether, dihydroartemisinin, then artemether, then artemisinin, artesunate. Then we have, I'm discussing about here the structure which I have given, artemether. Just see the structure. We have changed that lepton moiety. OCO, when we are having here OCO moiety, we have changed here with the OCH3. पहले हमने क्या किया था, डायहाइड्रो आर्टिमिसिलिन बनाया. डायहाइड्रो आर्टिमिसिलिन कैसे बनेगा, यहाँ पे एक हाइड्रोजन और एक OH, ठीक है. That means 2 hydrogen that has been added on this oxygen molecule. Here 1 hydrogen and 1 hydrogen on the O. So there is dihydro artemisinin. The structure is not shown here, but I have shown the structure with methyl group, or in combination we can say methoxy. So this is artemether. When I am putting here ethyl, then it is becoming arteether. So that means if we are putting dihydro artemisinin, which is good water soluble drug, whereas when we are putting methyl substituent, that is becoming good lipophilic drug. So artemether, arteether, they are highly lipophilic drugs as compared to artemisinin, which we have achieved that it is having poor water solubility or lipid solubility. By making the derivatives artemether, arteether, we can make our drug lipophilic, or by making dihydroartemisinin, we can make our drug hydrophilic. Fine. Now these are the marketed preparations of artemisinin, like with the tablet form, just we have shown for all the other drugs. And with this we finish all the classification related to the anti-malarial drugs, which I have discussed in detail, starting from every time whenever you have to start with the anti-malarial drugs, you have to start with the life cycle of Plasmodium. Until you will not explain the life cycle, then definitely the treatment regimen, that is not clear, because you should be very clear about the point that where your drugs are acting. So we studied about life cycle of Plasmodium, then we studied about its chemotherapy, 4 types of drugs, radical cures, whatever. And second, we have studied about the chemistry portion. In chemistry portion, we have studied with the detail about starting from the lead compound quinine. Then we did modifications. We got much and more potent compounds than quinine. So many drawbacks we discussed. And after that, we move on to 8 aminoquinolines, then 4 aminoquinolines, and then that is the bigonides, which is the less potent. I should not use the word least. It is the less potent drugs than. And I told you, because you have to give always the combination of anti-malarial drugs, so we would have some alternatives because of the emergence of resistance. And lastly, we discussed about artemisinin, which is very, very important. And with this, we finish this chapter, that is anti-malarial agents. Hope you would have understood all the things. Thank you.